Hello viewers, welcome once again to our ongoing series on Mendelism and beyond, and this is perhaps the last part of a discussion on epistatic interactions. If you remember, we had discussed on our last lecture about dominant epistasis and then again on a very interesting aspect of duplicate recessive epistasis, which we had nicknamed, or in fact Bateson and Punnett had called it as the complementary genes. Now moving ahead, let us now talk about the other two ratios which are still pending in terms of interallelic interactions with a few more examples. We first talk about the duplicate dominant genes. Now this again tells us that two interactions are involved. So that means there are in this duplicate type of gene interaction, there is a duplicate genes which are two pairs of alleles, whether they are alone or together, it does not matter. They are going to produce the same effect. Now in case of duplicate recessive epistasis, we were told that C and P together would produce a new anthocyanin containing phenotype. But here we say that these are two pairs of alleles, whether they are alone or they are together, the phenotype would be the same. So that means basically they are identical genes, but then they are situated on two different pairs of chromosomes. So that means in its own right, each gene is going to be dominant to its allele and it does not add to the effect of the other. So that means the genotype AA and the genotype AB would have the same intensity. So that means when these two pairs of genes are together, their phenotype is the same. When they are alone, it is again the same. Then when would the other gene express itself? The answer is very simple. If you can see the checkerboard a little later, we would find that the only option now that we are left with is that the recessive phenotype is going to express itself only when the gene pairs are double recessive. If they are not double recessive, then the character would prevail. So immediately it comes to our mind, even without looking at the checkerboard, that we are talking of just two phenotypic classes, and out of them just one situation of a double recessive exists in 1 by 16. So the other factor comes automatically by itself, and that is 15. So the ratio becomes 15:1. Let us illustrate this with the help of certain examples. As you can see on your screens, the first example happens to be of the fruit shape in shepherd's purse, a classical plant, Capsella bursapastoris, of the family, the mustard family Brassicaceae, which has earned a high name in our studies on embryology, in ultrastructure of eggs and fertilization, Capsella. Here we find that there are two types of morphologies of the fruits. The fruits are either triangular or they are ovate, and the genotype that we have given is capital T and capital V if they are together. You remember, I have just shown you two of these situations because these two morphologies are present, and the ovate morphology is going to be resorted to only when it is a case of double homozygous recessive. And on the checkerboard, immediately you will come to a conclusion that if you have a cross between a plant with triangular and an ovate fruit, the F1 progeny are all triangular fruit bearing, and in the F2 generation there is no combination which can spare itself from either one capital T or 1 capital V except 1. So the ratio automatically becomes 15 triangular and 1 ovate. So this is a ratio which talks about duplicate dominant epistasis. And on your screens, the classical picture which you would find in every book, every textbook is of the checkerboard of the triangular and the ovate fruits. So that means if you have other examples, we would find that the genes are a bit in a redundant system, and all those genes in a redundant system, they would have a duplicate gene action, and because they are encoding for the same product, or they are encoding products that at least they have a similar effect on a pathway. Let us consider another example of the flower color in snapdragons. As you would see on your screens, this snapdragon, that is Antirrhinum species, again belonging to the family Plantaginaceae, have two types of colors of flowers, and that is red and white. And the red ones would be signified by the capital A and capital B genes. Mind you, these are on two different chromosomes, but they are having the same effect, as I said in the beginning. And a flower would be white only when it is in a homozygous recessive, or let us say a double homozygous recessive condition. In the F2 generation, all the flowers naturally would be white, and in the next generation, that is F2, 15 combinations. You can see the abbreviated ratio also below this Punnett square would be having 15 red and just 1 out of 16 a double recessive phenotype. So this is the classical 15:1 ratio. If we were to explain this idea on a biochemical scale, again on your screens you would find that this trait is actually expressed wherever a dominant gene is present, fine, and that was 15 combinations, and one allele itself is capable of any anthocyanin production. So in the first box you would find that whether it is just A or the other case just B, you would have or AB together. The three situations seem to be identical as far as the conversion of the precursor into anthocyanin pigment is concerned. The only situation which is contrary is that neither of the capital A or B are present, and that would be just 1/16. So this is how we justify the ratio of 1:15. Now can you imagine, we sometimes wonder, and a student should always wonder, what would have happened if Mendel had worked on snapdragons to begin with? The results would have gone all awry. How could one get 9331 ratio? If you remember, in some of our earlier lectures on the history of Mendelism and the Mendelian principles, I had narrated to you the whole story of Mendel's work and his contradictions with Nageli and other people, and his working on Heretium, because Heretium had lot of linkages and other aberrations, so he never got the desired ratio of 9331. So the answer to the question which I just posed to you was that if Mendel had worked on snapdragons, perhaps we would have never had Mendelian principles. Taking forward the discussion, let us take a third example, and which is the endosperm color in maize. Now we find that in Zea maize the grains may be of two types. They could be yellow or they could be colorless, and in this case the yellow endosperm would again be an example or a product of either both the dominant alleles or either of them. So that means in any case yellow production would be there. And if you are going to make a cross between say Y capital ones Y1, Y1 and Y2, Y2, they will all be yellow, and all the recessive they would be having white endosperms. And if we then have the F1, all the progeny in the F1 would be yellow because the capital Y's are present, and the checkerboard in the F2 generation, if you see on your screens, would again be the same 15:1. So that means this is also a good example of the duplicate dominant epistasis. A very classical example that few people know is of the floating habit in rice. You know, the rice are also of two habits, either they float or they don't. So if we have and non-floating variety is dominant. So if we signify this variety by capital A and capital B, then we cross it with a floating one, we find that in F1 all the progeny, all the products are non-floating plants. And on your screens you can find the floating and the non-floating in the F2 generation being 15:1, 15 non-floating and just 1 out of 16 as the floating ones. So that means the basic idea of duplicate recessive state would be that it is only the floating habit in a homozygous recessive state which is going to produce a recessive coloration. The last part of our discussion now is going to revolve around, because we've already seen recessive epistasis, duplicate recessive epistasis, dominant epistasis, as well as the duplicate dominant or the double dominant epistasis. The only possibility which the genes could have is a dominant and recessive interaction, and this dominant and recessive interaction is going to have a totally different ratio, and it will also have its own type of inhibitory process. What really happens here is that a dominant allele at one locus can mask the expression of both the alleles at the second locus. Normally it was not doing like that. Normally a dominant allele was masking the dominant allele of the other, but here both the alleles are masked. So that means only two types of F2 phenotypes will result. A dominant genotype is present at one locus, let us say capital A, and the recessive genotype at the other, that is small b. They are going to produce the same phenotype effect. I would repeat, the dominant allele of one and the homozygous recessive allele of the other locus would produce the same phenotypic effect. Now again we would immediately rush to a conclusion without looking at the details of the abbreviated ratio or the checkerboard, that is only two types of phenotypic classes are possible in this situation. So that means one category of phenotypes would be, it contains one capital A and one capital B. The other category capital A and the other one. The other category would be small a, small b, because they have the same effect as the capital ones. They will produce just one type of phenotype, and the only case which remains is that BB is not homozygous recessive, it is only capital B and small b. So that would be a character. Let us see on the checkerboard with an example. The first example which we take here is with a chicken feather color. In chicken feather color we have two types of breeds, the white leghorns and the white Plymouth rocks. Now the white plumage of one of the chickens is dominant over the colored variety, whereas in the other, that is the white Plymouth, it is recessive to the colored variety. If you can see on your screens, we would have a white leghorn is dominant because there is an inhibitory factor which is present. Now, this inhibitory factor is not going to allow any color to develop on this chicken. So, I is the inhibitory factor and capital C is the color factor, and then of course, small I the recessive and small c as the recessive allele. When is the white color going to be produced? There are so many situations. Either both color and inhibiting factors are present. So, that means the genotype should have at least one capital I and one capital C, or a second situation would be that both the color and the inhibiting factors are absent. So, if there is small I and small c, how can color be produced, because there is no capital C? And the third situation is only the color factor is absent, that you have a situation of capital I and a small c together, or small c and small I together. In any case, the color of feathers of the chicken would remain white. Only when the capital C is present in the absence of capital I. And what would be that combination? Capital C, small I, and capital C, small I. That means there are only 3, and therefore the ratio immediately comes to your mind as 13:3. Let us prove this with the help of a cross and a checkerboard later. The white leghorn and white Plymouth Rock, both of them are white because both of them have at least one capital I, which is inhibitory. In the F1 generation, again all of them were white, and in the F2 generation, if you look very carefully, we have just 3 colored phenotypes. And what are the 3 colored phenotypes? As you can see on your screens, you would find that one of the colored ones has small I and small c. The other one has small I, one capital C and small c, and so is the genotype of the third one also. So, this ratio becomes 13:3. If we were to explain this whole concept again on a biochemical basis, we would find that a situation of capital C having just its own presence, we would have no pigment at all because capital I is present. If you have a situation of capital C and small I, fine, then we have a situation of a colored fowl or a colored chicken. And in the remaining 2 situations, we have again the white ones because capital I is not there, fine, but then capital C is also not there in the remaining 2 genotypes, which proves the point. The other example which we wish to signify here in terms of 13:3 ratio is the aileron color in maize. Now, the aileron layer in Caryopsis of maize is having a gene capital R, and if it is there, then the phenotype is red, and if it is small r, then the phenotype is white. There is again, like in the previous example, a second gene capital I. Now, this is going to inhibit the expression of R also, but it is not going to affect anything else. So, a situation of capital R and capital I definitely is going to be responsible for white coloration. And if you see on your screens the checkerboard, you would find that again we have what is called as the typical 13:3 situation, and one should also signify that why have we labeled the last of, that is 1/16 in red. It should have been in black. It is just to distinguish ourselves, to say that there is just one combination which is of a double homozygous recessive, but then it has the same morphology as the first one. So that means if we are going to summarize, we would find that 9 plus the third 3 plus the fourth 1 taken together, that means 9, 3 and 1, 13 would be white, and 3 in the third case would be the phenotype of the colored ones. So to summarize all these interactions, you can see on your screens a pictorial depiction of the different types of interactions, which are ranging from 9331, 9331 again, transforming to 9:7, and then the recessive that was complementary genes, and 934 of recessive epistasis, and the ones which we have discussed today in the form of 1231 and 15:1. We could also explain these ideas on a biochemical basis as to which genes are present and which would not allow a particular pigment to develop or will induce them to do so. So just to end this discussion, we have a slide which gives you again an overview of the modified dihybrid crosses as to how the expected Mendelian ratio of 9331 gets modified into various options. In viewers, in some of our next lectures, we would now like to take up a separate line of study, and that would be in terms of linkage, crossing over, and that's also very interesting aspect of the deviations from the normal expected Mendelian ratios. Thank you.